Hello everyone, very welcome to Vyas Edification. Today we are starting with a new series of NCERT discussion of biology, and in this series we are going to discuss all the questions given in your NCERT textbook. So first chapter we are taking in this series is a very important chapter and an interesting chapter that is human reproduction from class 12. This chapter contains 21 number of questions and we are taking all of those questions here. The first question we have is in the form of fill in the blanks. So first A is asking that humans reproduce asexually or sexually. Of course humans reproduce sexually. Humans reproduce through sexual means of reproduction. The next we have is humans are dash. Oviparous, viviparous or ovoviviparous. Of course, humans are viviparous, we are mammals, and we are viviparous. We give birth to young ones, thus we are known as viviparous animals. Third we have is fertilization is dash in humans. As we know that in the case of humans, fertilization takes place in the fallopian tube of the female body. Hence, the fertilization is internal in case of humans. So, it is internal. Fertilization is internal in humans. Next, we have is male and female gametes are haploid or diploid. As we know that gametes are always haploid. They contain a half set of chromosomes, that is 23 number of chromosomes in the case of humans. Thus, either the gametes from male or female, the gametes are always haploid in nature, containing only 23 number of chromosomes. Next we have as a zygote is diploid or haploid. We know that the gametes, male gamete and female gamete, when fused together and forms a unicellular structure, that unicellular structure is known as zygote. And zygote is diploid in nature because it is formed of fusion of two cells, formed of fusion of two gametes, that is male gamete and female gamete. The gametes are haploid. Both the haploid gametes when fused together they form a diploid zygote. So the zygote is actually diploid. Then next we have is the process of release of ovum from a mature follicle is called. We know that the process of release of ovum from ovary into the fallopian tube, there is a process known as ovulation. So this is ovulation. The process of release of ovum from a mature Graafian follicle is known as ovulation. Next we have is ovulation is induced by a hormone called ovulation is induced. Ovulation is the process of releasing of ovum from a mature Graafian follicle. That process of ovulation is actually induced by a hormone known as luteinizing hormone. LH, luteinizing hormone, is responsible for the process of ovulation. Next we have is the fusion of male and female gamete is called. The fusion of male and female gamete is known as fertilization. The fertilization is a process in which there is a fusion of male and female gamete takes place. Next we have is fertilization takes place in. As we know that fertilization is actually internal in case of humans. But where does actually the fertilization takes place? First of all, fertilization takes place in the female human body, but which organ is actually associated with the fertilization? Fallopian tube is the answer. In fallopian tube, the fertilization takes place. So the answer of this will be fallopian tube. Fallopian tube. Fertilization takes place in fallopian tube. Next we have is zygote divides to form dash, which is implanted in uterus. After the formation of zygote is a diploid structure which is formed with the fusion of female and male gamete. Then this zygote actually divides mitotically and undergoes to form a new structure which is known as embryo. But zygote, when transform into a new structure which is going to implanted in the uterine wall of a female, that structure is known as actually blastocyst. Blastocyst. Then we have the structure which provides vascular connection between fetus and uterus is called. The structure which provides connection between fetus and uterus is actually placenta, and placenta is vascular in nature. It is containing numerous blood vessels, hence it is vascular in nature, and placenta is the only organ or structure which connects the fetus and uterus of the female. The next question we have is, draw a labeled diagram of male reproductive system. Now let's make a male reproductive system. First we are going to make urinary bladder here. Then a gland known as prostate gland. This is urethra. Now we are going to make another gland over here, paired gland. This is seminal vesicle. Then we are going to make, first here are the ureters, tubes which conducting urine from the kidney into the urinary bladder, and here we are going to make accessory duct system of male reproductive system here. These are the ducts. Then we are going to make here a pair of testis which is present into the scrotal sac outside the abdomen. Now here next, in this testis we are going to make the compartments which is known as testicular lobules, inside which the seminiferous tubules are present. These seminiferous tubules present inside the testicular lobules will form a fine tube-like structures over here called retitestis. Then this retitestis will convert into a highly coiled longest tube of male reproductive system known as epididymis. Then this epididymis will go and form the tube known as vas deferens here. This is the complete male reproductive system. Now we are going to label this, and here also we have a foreskin like this. Now we are going to label this. This is testis. Now this is retitestis. Retitestis. These are testicular lobules inside which there are 1, 2, 3 seminiferous tubules. So these are testicular lobules. Testicular lobules. This portion, this swollen portion of penis is actually known as glans penis, And of course this is penis. This whole thing is penis. This whole thing is penis. So we can write here, this whole thing is known as penis, which is actually male external genitalia. Then here we can see a bulb-like gland over here. These glands are bulbourethral glands. Bulbourethral glands, also known as Cooper's gland. Then we have another gland here. This is the prostate gland. This is prostate gland. Now, these leaf-like structures you can see over here, these paired glands are known as seminal vesicle. Seminal vesicle. These are glands, of course. This is urinary bladder. This is urinary bladder. Now, this tube which is conducting urine from body to outside the body, this tube is known as urethra, and it also conducts sperms from testis and vas deferens, and the sperms will go here like this and comes out through this tube only. So, because of the path, following this path, the sperm over here, this is also known as, this tube is also known as urethra. This tube is urethra and also known as urogenital tract, as it is conducting sperms as well as urine. So this tube is known as urethra and urogenital tract. Now let's come towards the duct system. This is retitestis, and this is highly coiled tube of the male reproductive system and the longest tube also. So this tube is known as epididymis. Epididymis. And this tube, which is conducting sperms from the testis to the abdominal cavity, this tube is known as vas deferens. Vas deferens. So this is your male reproductive system, and of course here we can see a fold of skin over the penis, which is covering penis partially. So this skin, you can say this skin is foreskin. Now we have completed with the male reproductive system. So the next question, question number 3 we have is, draw a labeled diagram of female reproductive system. Now let's make female reproductive system. As we know that female reproductive system is consisting of a pair of ovaries, a pair of fallopian tubes and a uterus. So first we are going to make a uterus. This is uterus, which is inverted pear shaped. This is cervix, and finally we have vaginal canal. Now this portion, this swollen portion you can see is the fundus part of uterus. From here a fallopian tube arises like this, and the finger-like projections at the end of the fallopian tubes. Now we are going to make a pair of ovaries here. Ovaries. Which is connected to the pelvic wall of our body and as well as to the uterus with the help of ligaments. The help of ligaments. So these are the ligaments which are connecting the ovaries to the uterus as well as to the pelvic wall of our body. Now we are going to make the layers inside the uterine cavity. So this is a structure of female reproductive system. Now we are going to label this diagram. As we know, this is ovaries. So this is ovary, which always occur in pairs. This is fallopian tube, a pair of fallopian tube we have in our female reproductive system, but we have different parts or compartments in the fallopian tube. As we can see, the touching portion of finger-like projections of the fallopian tube, which actually collects the ova from ovary and it conducts to the fallopian tube, inner parts of fallopian tube. This finger-like projections of the fallopian tube is known as fimbriae. So these finger-like projections are fimbriae. Then we have a swollen region inside the fallopian tube. This is ampullary region, or we can call it ampulla. Ampulla. Then we have next region which is close to the uterus. That region of fallopian tube which is in the close region to the uterus is known as isthmus. So I am writing here, this portion is known as isthmus. So this is the labeling of fallopian tube we have completed with. Now let's come towards the uterus, which is single in number, and this swollen region on the upper side of the uterus, you can see this is the fundus part. This is fundus part of uterus. This is the uterus body of uterus, and inside which we have a cavity in which the baby actually resides. This cavity is known as uterine cavity. Uterine cavity. Below this you can see this is cervix, and here we have is vagina. So we are completed with the labeling of a female reproductive system. Last thing which is remaining in this diagram is the labeling of walls of uterus. So from outside to the inner sides we are having 3 layers of uterus, and the outermost layer of the uterus is known as perimetrium. Perimetrium, which is a protective layer, inside which the middle layer we have is myometrium. Myometrium, which contributes in the contractions during labor. Then inside we have the innermost layer of uterus, that is endometrium. Endometrium, which undergoes cyclical changes and constitute pregnancy as well as menstruation cycle. So this is the complete labeled structural diagram of female reproductive system. The next question, question number 4, is write 2 major functions each of testis and ovary. So let's discuss the functions of each of testis and ovary. Here we are writing the functions of testis. So testis, we know that is a male primary sex organ. So being a male primary sex organ, it provides the site, for the production of sperm cells or male gametes. We know that testis is a primary male sex organ inside which there are semifirst tubules inside testicular lobules. Seminiferous tubules are having specialized cells known as spermatogonia. These spermatogonias are actually giving rise to sperm cells or male gametes. So this is the first function of testis, that is, it provides the site for the synthesis or production of male sperm cells or male gamete. The next second function of testis is, we know that if we talk about the testis here, testis is having testicular lobules, 250 compartments known as testicular lobules. Testicular lobules are having 1 to 3 fine tubes, and these tubes are known as seminiferous tubules. Now you can see the space inside the testicular lobules outside the seminiferous tubules. This space in testicular lobule is known as interstitial space, and inside this interstitial space we have a specialized cells known as interstitial cells, or we can call them Leydig cells. These Leydig cells present inside the interstitial space of a testicular lobule actually produces a hormone known as testosterone, which is very essential for the secondary sexual characters in male. So testis is also having another function, that it helps in the production of testosterone, which is of course very essential hormone for the secondary sexual characters in the male. Now let's come towards the functions of ovaries. Now if we talk about the ovaries are also the female primary sex organs. If we talk about the functions of ovaries, first function of ovaries is it provides the site for the production of ovum or ova. Of course, being a female primary sex organ, it also provides the site for the production of ova, which is a female gamete. We know that the production of ova, or we can call oogenesis, this oogenesis takes place inside the ovaries. So this is the first function of ovaries. Next we have is it actually produces two types of hormone, that is estrogen and progesterone. These two hormones are actually produced by the ovaries only and are essential for secondary sexual characters in the females. So this is the functions of each of the testis and ovaries. The next question we have is, describe the structure of a seminiferous tubule. Now, what is seminiferous tubule? We know that these are the fine tubules inside the testicular lobules of a testis in the males, and seminiferous tubules are having specialized cells which are forming the male gametes or sperm cells. Now, if we talk about the testis, we know that this is testis and it is having 250 compartments like this. 250 compartments known as testicular lobules, inside which there are 1, 2, 3 seminiferous tubules are present like this. If we cut the testis and see the structure of seminiferous tubules, let's make a whole circle which is representing a testicular lobule, inside which we know that inside the testicular lobule we are having 1 to 3 seminiferous tubules. Let's say we are having 2 seminiferous tubules inside this particular testicular lobule. So this, first of all, we are going to mark this. This is testicular lobule. This is testicular lobule, and inside which this space, you can see this space is interstitial space, which is having specialized cells known as interstitial cells or Leydig cells. So this space is known as interstitial space. Now these circles inside the testicular lobules, you can see these are seminiferous tubules. So these are seminiferous tubules. Now, we are going to describe the structure of a seminiferous tubule. Let's focus on this particular structure. This is seminiferous tubule. Now, inside the seminiferous tubule, the inner lining of seminiferous tubule is actually lined by two types of cell. This inside surface of seminiferous tubule are lined by two types of cells. Let's say these are the first type of cells lining the seminiferous tubules from the inner surface or inner side. These particular cells are known as spermatogonias, and also there are another type of cell. Let's make them like this. These another type of cells lining the seminiferous tubules, these are Sertoli cells. So inside surface of seminiferous tubule is actually lined by the two types of cells, that is spermatogonia and Sertoli cells. These are spermatogonia and these are Sertoli cells. These spermatogonia cells are actually going to form the sperm cells or male gametes, and the another type of cells present inside the seminiferous tubule is actually the Sertoli cells, and they will provide the nutrition for developing sperm cells inside this seminiferous tubule only. After the formation of sperms or sperm cells inside the seminiferous tubules, they are released into the lumen of the seminiferous tubule, from where they are going to conduct through duct system of the male reproductive system. So this is the structure, detailed structure of a seminiferous tubule. Now let's move towards the next question, that is, what is spermatogenesis, and briefly describe the process of spermatogenesis. First of all, the first part of this question is asking what is spermatogenesis. Now, spermatogenesis is actually a process through which the formation of male gamete or sperm cells takes place. So the process through which the sperm cells are being formed, this process is known as spermatogenesis. Spermatogenesis takes place inside the testis, and precisely if we say inside the seminiferous tubules, as the seminiferous tubules are having specialized cells known as spermatogonia, and these spermatogonias are going to give rise to sperm cells or male gametes. So the spermatogenesis is a process of formation of male gametes or sperm cells inside the testis of a male. We have completed the first part of this question. The second part of this question is asking briefly describe the process of spermatogenesis. So let's describe the process of spermatogenesis with the help of a diagrammatic representation. Here we are taking spermatogonia. This is spermatogonia. Spermatogonias are present inside the seminiferous tubules of the testis in the males. So we are taking spermatogonias because these are the cells which actually give rise to the sperm cells or male gametes. So this is spermatogonia, which is undergoing the process of mitotic division. It is undergoing mitotic division or mitosis. Now, why is it undergoing the mitotic division and why not meiotic division? Because the spermatogonias wants to multiply, they want to increase in number. So, for increasing number, they are undergoing the mitotic division. Now, after completing the mitotic division, there will be several cells. Now, these cells will actually differentiate or grows and forms a huge cell like this. This is primary spermatocyte. See here, spermatogonia is undergoing the mitotic divisions to multiply or to increase in number. After this, when we will have a huge number of spermatogonias, among these all spermatogonias, some of them will grow in size, will differentiate and transforms into a new cell. That new cell is known as primary spermatocyte. This is primary spermatocyte. Now, This primary spermatocyte will undergo the meiotic division, and as we know that meiotic division will occur in two phases, meiosis 1 and meiosis 2. So first of all, this primary spermatocyte I am drawing here, I am writing here, this primary spermatocyte is undergoing the first meiotic division, that is meiosis 1. This primary spermatocyte is undergoing meiotic division 1 and producing two cells. There is a equal division is happening. So primary spermatocyte is undergoing the first meiotic division and is forming two cells. These two cells are known as secondary spermatocytes. Secondary spermatocytes. Now, after completing meiotic 1 division, each of the secondary spermatocyte will undergo the second meiotic division. So both of the secondary spermatocyte will undergo the second meiotic division here, second meiotic division, and after completing the second meiotic division, first of all I am writing here that they are going second meiotic division, which results into the formation of 4 cells over here, 4 cells. These 4 cells after completing the meiotic division are known as spermatids. Now, these all spermatids will differentiate into a new structure and they will form sperm cells or spermatozoa. They will form sperm cells or spermatozoa. So this is spermatogenesis, that a spermatogonia will undergo mitotic division to increase number of them. Then among all these spermatogonia cells, some will transform into primary spermatocytes like this. Primary spermatocyte will undergo meiotic division, one meiotic division, and forms 2 cells, which is secondary spermatocytes. Each of the secondary spermatocyte will undergo second meiotic division and forming 4 cells known as spermatids. Each of the spermatid will differentiate into a new structure known as sperm cell or spermatozoa. Now let's discuss the ploidy level of each of the cells written here. Now, the spermatogonia, which is the very first cell of in the process of spermatogenesis, the spermatogonia is 2N, diploid cell, which is a part of our body only. So this is diploid cell. Now it is undergoing the mitotic division or equational division. So there will be no reduction of chromosomes in them. So the spermatogonias which are increased in number will always be 2N. All these are 2N. As you can see, there is no division is occurring from here to here. So first cell, the primary spermatocyte are always 2N. These are also 2N. These are also diploid cells. Now let's come towards this. This is also a primary spermatocyte being diploid. This is undergoing meiotic division to form sperm cells or male gametes. So when any cell is undergoing meiotic division, there will be the reduction of chromosomes by half. So as this primary spermatocyte is undergoing first mitotic division producing 2 daughter cells, so these cells will be haploid. So primary spermatocyte undergoing first meiotic division will result into the formation of 2 haploid cells, and these 2 haploid cells are known as primary spermatocytes. These primary spermatocytes are undergoing second meiotic divisions. So all these cells known as spermatids are also haploid. All these spermatids are haploid, and thus all these spermatids are just transforming. They are not undergoing any kind of division. These all spermatids are transforming into new structure known as sperm cells or spermatozoa. So all these spermatozoa are always haploid. These are also haploid. Hence proved that all gametes, either male or female, are always haploid. As we can see, the process of spermatogenesis finally resulting into the formation of spermatozoa or sperm cells are haploid. You can see these are haploid cells or haploid gametes. So we have completed with the process of spermatogenesis and what is actually spermatogenesis. Now next question we have is question number 7, that is name the hormones involved in regulation of spermatogenesis. So basically in this chapter we always confuse with the hormones. So let's make it clear that the hormones which are actually involved in the regulation of spermatogenesis in the case of males, as the spermatogenesis is the production of sperm cells or male gamete, of course it will happen in the case of males. So if we talk about the hormones in males, there are actually 3 hormones in the males in the reproduction process. That is, we have FSH, follicle stimulating hormone. Then next we have is LH, luteinizing hormone, and the third hormone we have is testosterone. Testosterone. Now let's discuss all of the functions. First of all, if we talk about the FSH is having a function that FSH, follicle stimulating hormone, is actually stimulating the Sertoli cells to provide nutrition for the developing sperm cells or male gametes. So FSH is actually stimulating the Sertoli cells to provide the nourishment for the developing sperm cells. Next we have is luteinizing hormone, LH. Luteinizing hormone stimulates the Leydig cells. Leydig cells, we know that Leydig cells or interstitial cells are present inside the interstitial space of testicular lobules. So LH, luteinizing hormone, actually stimulates the Leydig cells to synthesize a hormone known as testosterone. And testosterone is the hormone which is actually primarily involves in the regulation of a process of spermatogenesis. So it maintains spermatogenesis as well as secondary sexual characters. Secondary sexual characters in males. So here we have the functions of all these hormones, that FSH, follicle stimulating hormone, is actually stimulating the Sertoli cells to provide the nourishment for the developing sperm cells or male gametes. Hence it is actually, that is follicle stimulating hormone is actually indirectly helping or regulating the process of spermatogenesis. Next we have is Leydig cells. Leydig cells stimulates the Leydig cells. Luteinizing hormone actually is stimulating the Leydig cells for the synthesis of testosterone. Hence this is also indirectly is involved in the process of spermatogenesis. But the main hormone, the primary hormone, important hormone which is actually directly involving in the process of spermatogenesis, that is testosterone. So testosterone is the primary hormone. Testosterone is the important hormone which directly involves in the regulation of spermatogenesis. So here we have answer of this question. Next, question number 8, we have define spermiogenesis and spermation. So this is also a question in which always we get confused that what is the difference between spermiogenesis and spermiation. So let's have a difference between these two. Here we are writing with the spermiogenesis. Spermiogenesis is a process through which Spermatids transform or differentiates into spermatozoa or sperm cells. So spermiogenesis is a process, the process by which the spermatids gets transformed into spermatozoa or sperm cells. So this is the process of transformation of spermatids into spermatozoa or sperm cells. This process is known as spermiogenesis. There is another process known as spermatogenesis. Spermatogenesis is the process of formation of male gamete or sperm cell. Spermatogenesis is that. Now we are having spermiogenesis. Spermiogenesis is a process by which spermatids are transforming into spermatozoa or sperm cells. The next we have is spermiation. Spermiation is a process in which after the formation of spermatozoa or sperm cells, they are being released into the lumens of seminiferous tubules. Just remember one thing, that we have seminiferous tubules like this. Seminiferous tubules are lined by two types of cells from the inner surface. One is spermatogonia and another one is Sertoli cells. Now, this is the lumen of seminiferous tubule. We know that the formation of spermatozoa or sperm cells are actually taking place inside the seminiferous tubules because the spermatogonia cells or the male germ cells are present inside the seminiferous tubules only. So these spermatogonia cells will give rise to the spermatozoa or sperm cells inside this. These spermatozoas are actually embedded into the Sertoli cells to suck the nutrition from these Sertoli cells. After the maturation, after they get mature, these sperm cells are released into the lumens of Sertoli cells. We know that this spermatogonia will give rise to the sperm cells. These sperm cells will remain embedded into the Sertoli cells to get nutrition from them. After they get mature, they are being released into the lumen of the Sertoli cell here. They are being released into the lumens of Sertoli cells. This process only, this process of releasing of sperm cells or spermatozoas from Sertoli cells into the lumen of seminiferous tubule, this process is known as spermiation. So I am writing here that release of spermatozoa into the lumen of seminiferous tubule. This process is known as spermiation. So this is the basic difference between spermiogenesis and spermiation. Spermiogenesis is a process in which spermatids are being transformed into the spermatozoa or sperm cells, and spermiation is the process in which the sperm cells or spermatozoa, after getting mature, they are being released into the lumens of seminiferous tubules. So this is the answer of this question number 8. Next, question number 9 is draw a labeled diagram of sperm. Another diagram we have. So let's make a diagram of sperm here. Now, human male sperm looks like this. This is head of a sperm. As we know that a human male sperm is actually consisting of 3 parts, that is head, middle piece and a tail. So we are first making the head of a sperm. This is the head of a sperm. Then the small portion is actually a neck of the sperm, and then we have a middle piece. And we have a middle piece like this, and after this middle piece actually is forwarding into a tail. Now, this head region is actually having a cap-like structure over here. This cap-like structure over here. Here we have a centriole and numerous mitochondria in the middle piece of a sperm, like this. This is filament inside the tail of a sperm. This whole structure of sperm or a sperm cell is actually lined by a plasma membrane, like this. The plasma membrane is lining the whole sperm. So this is the structure of sperm. Let's label this diagram of a sperm cell. So first of all, this region, this whole region is actually a head of sperm. So this is a head of sperm containing nucleus. A haploid set of chromosome is present inside the nucleus of a sperm cell, as it is a gamete, as it is a male gamete. So this is head containing a haploid nucleus, a haploid nucleus. Then this portion, the upper portion, cap-like portion or cap-like structure on the head we have is acrosome. This is known as acrosome, which is containing hyaluronidase enzymes for the penetration of egg at the time of fertilization. So this is acrosome, which is having certain types of enzyme. Then we have head region. Then we have, this is our middle piece. This is middle piece. Middle piece, inside which there are numerous mitochondria are present. Mitochondria are present, which are present in the spiral form. There are numerous mitochondria present inside the middle piece, which actually provides the energy to this sperm cell. Then this portion, this whole portion, you can see this is a tail which provides motility to the sperm cell. This filament you can see inside the tail is actually facilitating the motility property inside the sperm cells or spermatozoa in the males. This is a complete labeled diagram of a sperm cell. Next question number 10 we have is, what are the major components of seminal plasma? Now let's discuss this. Seminal plasma along with sperm cells or sperms constitute together to form a mixture, and this mixture is known as semen. So we have semen. Semen constitute two components, that is sperm cells, which are actually made inside the testis, and another portion in the semen we have is seminal plasma, which is a mixture of three glands associated with the male reproductive system. As we are discussing the major components of seminal plasma is actually a mixture coming from the three glands of the male reproductive system, and in the case of male reproductive system we have three types of accessory glands. What are these glands? First gland is seminal vesicle. Seminal vesicle, the number one gland we have is seminal vesicle. This seminal vesicle gland is actually present in a pair. So we have two seminal vesicles in the male reproductive system. Now let's talk about the contribution of seminal vesicle. Seminal vesicle contributes about 60% of the seminal plasma. First of all, this portion, the mixture coming from the seminal vesicle is actually contributing 60% in the seminal plasma. This seminal vesicle is providing us with the fructose, which is of course a nutrient. Then we have mucus. Then we have some enzymes. And it also provides alkaline pH, which counteracts the acidity in the male urethra as well as in the vagina of a female. So this is the contribution coming from the seminal vesicle. It contributes about the 60% of the seminal plasma. It provides us with the fructose, which is a nutrient. Then it provides some mucus and some certain enzymes, and also it provides alkaline pH, which counteracts the acidity in the male urethra as well as in the vagina of a female. Next gland, number 2 gland we have is prostate gland. Prostate gland is actually 1 in number. This is not a paired structure, this is 1 in number, prostate gland. If we talk about the prostate gland contributes about 30% of the seminal plasma. 30% of the seminal plasma is actually contributed by the mixture coming from the prostate gland. What does the prostate gland provide? Prostate gland provides citrate. Prostate gland provides citrate, which is a nutrient for mitochondria present in the middle piece of a sperm cell. So this is the energy source. Citrate is the energy source, or we can say through this citrate the mitochondria is actually make energy. Then after citrate, prostate gland also provides us with calcium ions and also with proteolytic enzymes, which helps in the liquefying of the ejaculate just before releasing. So prostate gland is actually contributing the 30% of the seminal plasma. It provides us with the citrate, calcium ions and proteolytic enzymes. The third accessory gland in the male reproductive system we have is bulbourethral gland. Actually, this bulbourethral gland provides alkaline pH. This gland does not contribute much to the seminal plasma. This gland is actually lubricates the passage for semen, and it also have small function in the seminal plasma, that it contains buffer just like the seminal plasma, and it helps in the counteraction of acidity inside male and female. So these are the components or contributions of seminal plasma. Next question number 11 is asking what are the major functions of male accessory ducts and glands. We have asked with the functions of male accessory ducts and glands. So if we talk about the male accessory ducts, first of all, male accessory ducts constituting the ducts like vas deferens. Then we have epididymis. We have vas efferentia and retitestis. All these ducts, vas deferens, epididymis, vas efferentia, retitestis, all these ducts constituting together to form a duct system in the male reproductive system, and the function of all these accessory ducts is just to conduct the sperms or spermatozoa from the abdominal cavity, to the abdominal cavity, so as they can pass the sperm cells outside the body, inside the female reproductive tract. So this is the function of accessory ducts. Let's have the function of accessory glands. As we have discussed in the previous question that the accessory glands named as seminal vesicles, prostate gland and bulbourethral gland, all these glands actually produces mixtures of some substances, and all the mixture coming from all these accessory glands of the male reproductive system is forming seminal plasma, which is a part or constituent of the semen. So here are the functions of accessory ducts, and we have discussed the functions of male accessory glands. Functions of male accessory glands such as seminal plasma. Seminal plasma is actually providing us with the huge portion of nutrient, that is fructose, and it also provides us with the acidic pH, the alkaline pH, which counteracts the acidic pH of vagina, and some enzymes. The prostate gland of male reproductive system is providing us with the proteolytic enzymes, citrate and calcium ions, and the bulbourethral gland, as it is containing buffer solution, so it also provides us with the alkaline pH. So these are the functions of male accessory glands as well as the male accessory ducts. Next, question number 12 is, what is oogenesis? Give a brief account of oogenesis. So first of all, what is oogenesis? Oogenesis is a part or is a process of formation of ovum or formation of egg cell inside the ovaries of a female. So oogenesis have a simple definition, that is, it is just a process by which the formation of ovum or the formation of egg cell takes place inside the ovaries of a female. Now let's understand the account of oogenesis, a brief account of oogenesis with the help of a diagrammatic representation. First of all, you have to remember that oogenesis is actually different with the spermatogenesis, as the oogenesis starts before the birth of a female child. So oogenesis, a very important point to remember here is, oogenesis actually starts even before the birth of a female child. So here we are writing, this is fetal stage of a female child. All these things which are writing over here on the screen are actually occurring in the ovaries of a female. Now in the fetal stage we have many female germ cells known as oogonias at the stage of a fetal. At the time of when the female child is present inside the womb of her mother, that time is she is having around 6 lakhs of oogonias, or we can say 6 lakhs of follicles, primary follicles, inside which the oogonias are actually present. Basically this oogenesis is somewhat related to the folliculogenesis. So we are taking oogenesis here. This oogenesis, the formation of egg cell, the formation of ovum, is actually happening inside the ovaries of a female, and this oogenesis or the formation of egg is occurring inside the follicles only which are present in the ovaries. Now this oogonia divides mitotically. This divides mitotically. This will undergo mitosis. This will undergo mitosis, and first of all this oogonia is actually diploid in nature. Now these oogonias in the fetal stage will undergo mitotic division and form several cells. Now we will have cells like this. Among all these cells, after multiplication, after the mitotic division, when this oogonia undergoing mitotic division to increase their number, among all these cells, several cells will transform into a cell, a grown cell which is known as primary oocyte. This is primary oocyte. Now this primary oocyte will undergo first meiotic division. First of all, This is as there is no meiotic division is occurring, hence the primary oocyte will also be diploid in nature. Now at the time of fetal stage, only this primary oocyte will undergo the first meiotic division, but it will not complete the first meiotic division. It will remain suspended in the diplotin stage of prophase 1 of meiotic 1 division. So it starts meiotic division, I am writing here, it starts first meiotic division in the fetal stage only. It will start the first meiotic division into the fetal stage only, but remain suspended in the diplotin stage, of prophase 1. It is not completing the first meiotic division. While it is getting into the first meiotic division into the fetal stage only, but it is not capable of completing this division. It will remain suspended in the diplotin stage of prophase 1 here. Now, this all thing is happening in the fetal stage of a female child. When the female child takes birth, suppose the female child is now taken birth, this will remain same up to the puberty. This will the stage from here to here, this will the stage called as childhood. Now once the this female child after taking birth and completing the childhood, when hits the puberty, let's suppose this female child hits the puberty. Now this first meiotic division is going to be completed at the time of puberty only, first puberty. Now the fetal stage is having oogonia cells which undergoes mitotic division to form primary spermatocytes. Primary spermatocytes in the fetal stage only, they will start the first meiotic division but is not capable of complete the first meiotic divisions, hence they will remain suspended in the diplotin stage of prophase 1 and they will complete the first meiotic division when this female child is going to hit the puberty. At the time of puberty, this first meiotic division will going to be complete at this stage. But here to remember is that this first meiotic division will result into the formation of two unequal cells. Hence, this meiotic division will be unequal division. Now, at the time of puberty, this primary spermatocyte undergoing or completing the first meiotic division will result into the formation of two unequal cells. Now, we have two unequal cells like this. This smaller cell is known as first polar body, and the bigger one cell we have is secondary oocyte. This is secondary oocyte. This thing, after the completion of first meiotic division, when the formation of first polar body and secondary oocyte is being takes place, this is happening actually in the phase of puberty. When the female child hits puberty, we will have first polar body and secondary oocyte. This secondary oocyte will undergo another meiotic division, the second meiotic division. Now this secondary oocyte will undergo meiotic division, but it will also remain suspended in the metaphase 2. So this secondary oocyte will remain suspended in the metaphase 2. It will also not complete the meiotic division. It will also not complete the second meiotic division. It will complete the second meiotic division only when the sperm cell will come in contact with the secondary oocyte. When the ovulation occurs inside the females, ovulation means the release of these secondary oocytes from the mature Graafian follicles from ovaries into the fallopian tubes. This is known as ovulation. When ovulation is being done, and inside the fallopian tube, when this secondary oocyte actually meets with the sperm cells, let's suppose the sperm cell came to the secondary oocyte. This sperm cell, when actually meets with the secondary oocyte, it activates this secondary oocyte. Hence, the completion of second meiotic division will take place and resulting into another unequal cells, that is, we will have small cell and a big cell. This small cell is known as second polar body, and this one big one cell we have is ovum. Now, in all the females, when we are talking about the secondary oocyte, in all females there is no formation of ovum actually takes place. Only the formation of ovum takes place when the secondary oocyte meets with the sperm cells inside the fallopian tube. Otherwise, when the secondary oocyte is not meeting the sperm, this secondary oocyte will release into the fallopian tube, then it will go into the uterus and will come outside of the body through the vagina during the menstruation period. So this is how oogenesis actually takes place inside the into the fetal stage. Oogonias divides mitotically to form primary oocytes. Primary oocytes are 2 and diploid. This primary oocyte starts its first meiotic division but is not capable of completing it. Hence, it remains suspended in the diplotin stage of prophase 1. And when this female child hits the puberty, it will complete the first meiotic division, forming the two cells. These two cells, all these two cells, the first polar body and the secondary oocyte, which are unequal in size, are having N chromosomes, are having haploid ploidity. So all these cells, that is first polar body and secondary oocyte, are haploid in nature. Then after this, secondary oocyte will also undergo the second meiotic division but is also not capable of completing it, thus remain suspended in the metaphase 2. This will complete the meiotic 2 division only when it will meet the sperm cell inside the fallopian tube. If the sperm cell gets meet with the secondary oocyte, this secondary oocyte will complete the metaphase 2 and goes into anaphase 2, completing the meiotic 2 division and converting into an ovum and second polar body. These secondary polar body and first polar body actually degenerates. What we will get finally? The ovum, which is also haploid in nature, containing 23 chromosome numbers. So this is the process of oogenesis, which is actually starts much before the childbirth or female childbirth, fetal stage, and continues till here, puberty. Next, question number 13. Draw a labeled diagram of a section through ovary. Now let's make a diagram of ovary here. Here we have ovary. As we know that inside the ovary, this whole space is actually known as stroma, and the stroma of ovary is actually divided into two compartments or zones. The peripheral zone of the ovary is known as cortex, and the inner portion of the stroma of an ovary is known as inner medulla. Now let's talk about the cross section of ovary. Ovary is having oogonias or primary follicles here. Primary follicles are actually having oogonias inside them. Oogenesis and folliculogenesis actually happen simultaneously. When we talk about the oogenesis, that is the production of egg cell or ovum, it takes place inside the follicles only. When the follicles matures, they grows inside the follicles only there the formation of ovum takes place. Now, if we talk about the section of ovary, we are having primary follicles here. These primary follicles will grow and convert into primary follicle here, then secondary follicle and tertiary follicle. Here we have is primary follicle, inside which the oogonia cells are present, which are transforming into the egg cell or ovum. Let's make here a tertiary follicle over here. Tertiary follicle, which is having our secondary oocyte granulosa cells, then a fluid filled cavity over here, the fluid filled cavity. The fluid filled cavity in yellow color you can see. This is a fluid filled cavity showing antrum. So we can mark this as the tertiary follicle. Of course, the tertiary follicle is made through secondary follicle. The primary follicle will transform into secondary follicle, secondary follicle will grows into the tertiary follicle. This tertiary follicle is characterized by having a fluid filled cavity, that is antrum. This cavity is antrum. So tertiary follicle is showing antrum. Then we have a mature follicle, that is Graafian follicle. That is Graafian follicle containing our oocyte. That is, this is your matured Graafian follicle. We will mark it as Graafian follicle. Now, when this Graafian follicle matures, this secondary oocyte will released from this Graafian follicle after rupturing of this Graafian follicle, and the secondary oocyte will goes into the fallopian tube. So this Graafian follicle here ruptures like this. This Graafian follicle ruptures here, and the secondary oocyte is being released from here. This is ruptured Graafian follicle. This ruptured Graafian follicle will remain inside the ovary and transform into another structure known as corpus luteum. Known as corpus luteum. This is corpus luteum. This is corpus luteum. Corpus luteum is formed from the remaining part of the Graafian follicle, which is being ruptured to release the secondary oocyte from ovary into the fallopian tube. This Graafian follicle will transform into the corpus luteum, which acts as an endocrine gland and produces progesterone hormone, which maintains the endometrium lining and uterus, which makes the uterus suitable for the pregnancy, possible pregnancy. If after ovulation the secondary oocyte does not meet with the sperm and the fertilization does not take place, this corpus luteum will degenerate. This corpus luteum will degenerate like this, and this degenerated corpus luteum will be called as corpus albicans. Corpus albicans. Now, inner medulla of the ovary is having nerve fibers and blood vessels. Nerve fibers and blood vessels. So this is the complete structure or complete section through ovary. The next question, question number 14 is, draw a labeled diagram of a Graafian follicle. Another diagram we have, let's make a diagram of Graafian follicle, which is a mature follicle through which, after rupturing of this Graafian follicle, secondary oocyte is being released from the ovary into the fallopian tube, where the fertilization takes place when this secondary oocyte actually meets with the sperm cell. Now, Graafian follicle looks like this. This is Graafian follicle. Graafian follicle is having granulosa cells like this. Granulosa cells. Now, this graphene follicle is actually also containing our secondary oocyte, which is not being completed its second meiotic division. So inside the graphene follicle we have an secondary oocyte here. This is our secondary oocyte, which is haploid and have not been completed its meiotic division. Secondary oocyte having a layer surrounding it. This layer is known as proteinaceous layer. First of all, this proteinaceous layer is actually known as zona pellucida. This is known as zona pellucida. Now, surrounding this zona pellucida, there is a layer of granulosa cells, multi-layer of granulosa cells surrounding the zona pellucida, and this layer is known as corona radiata. Corona radiata. Now, here also we have numerous cells of granulosa cells. Now, this is the structure of graphene follicle, and this cavity, this space you can see here in the graphene follicle, this cavity is known as antrum, which is filled with the fluid. This is the antrum or fluid filled cavity. Now this secondary oocyte is having zona pellucida, corona radiata and granulosa cells. This is the complete structure of a graphene follicle. After this maturation of a graphene follicle, this graphene follicle ruptures and releases the secondary oocyte from the ovary into the fallopian tube. And also the most important thing to remember is this secondary oocyte is actually present not in the center of this graphene follicle. It is actually present eccentrically towards the periphery of the graphene follicle. So this is the complete structure of a graphene follicle, you can see. Next, question number 15. Name the functions of the following. We have corpus luteum, endometrium, acrosome, sperm tail and fimbriae. We have to mention the functions of each of them. Let's talk about the very first thing, that is corpus luteum. If we talk about the function of a corpus luteum is a structure or an endocrine structure which is formed from the ruptured graphene follicle inside the ovaries of a female. And as it is known as endocrine structure which is pertaining inside the ovaries of a female, this endocrine structure or a corpus luteum is actually helps in the production, or it actually produces the hormone which is known as progesterone. Progesterone hormone is produced by this corpus luteum inside the ovaries. This progesterone hormone actually maintains the uterus for the possible pregnancy. So this is the function of corpus luteum, that it produces the hormone progesterone inside the female. Then let's move towards the next, that is endometrium. Endometrium is the innermost layer or innermost lining of the uterus, and it undergoes cyclical changes which result into the menstruation cycle, and it also helps in the pregnancy. So endometrium lining undergoes cyclical changes, which actually serves us as menstrual cycle or menstruation. In that menstruation cycle, menstruation when occurs, this menstruation occurs by this endometrium only, and also this endometrium maintains the uterus during pregnancy. So this is the function of endometrium lining of the uterus. Next we have is acrosome. Acrosome is actually filled with some special type of enzymes. that is hyaluronidase enzymes, and the enzymes present in the acrosome helps in the fertilization process of, here we can write this, acrosome is having enzymes. This enzyme helps the penetration, helps in the penetration process of fertilization. So this is the function of acrosome, that it is having certain types of enzymes in it, and these enzymes actually penetrates the ovum to fertilize that cell. Now next we have a sperm tail. Sperm tails actually facilitates the movement or motility to the sperm cell, movement to the sperm cell. So this is the function of sperm tail. Next we have is fimbriae. You all remember that what is fimbriae? Fimbriae is the last part or finger-like projections of the fallopian tube in the female reproductive system. Now what is the function of these fimbriae or these finger-like projections of the fallopian tube? These fimbriae or finger-like projections of the fallopian tube actually collects the ovum, not ovum, it actually collects the secondary oocyte from the ovary and it transports the secondary oocyte into the fallopian tube. So fimbriae have a function of collecting the secondary oocyte from the ovaries and it transport into the fallopian tube. So these are the functions of all these corpus luteum, endometrium to the fimbriae. Next question, identify true or false statement. Correct each false statement to make it true. First of all, we have to identify the statement whether it is true or false, and if the statement is false, we have to make it correct or true. So first statement given is androgens are produced by Sertoli cells. Androgens are produced by Sertoli cells. Testosterone, basically androgens are testosterones. Testosterone or androgen, which is a male hormones, and these male hormones are not produced by Sertoli cells. So the statement given is false, as androgens are produced by Leydig cells. We are correcting this statement. These are produced by Leydig cells. Leydig cells. Second statement given is spermatozoa get nutrition from Sertoli cell. This statement is very correct, that spermatozoa or sperm cell is actually getting nutrition from the Sertoli cell only. So this statement is true. Next is Leydig cells are found in ovary. No, Leydig cells are not found in ovary. Leydig cells are the part of male reproductive system first of all, and they are found in the interstitial space of the testicular lobules. So these are found in the interstitial space of the testicular lobules. Next statement we have is Leydig cells synthesize androgens. Yes, this is a true statement. This is a correct statement, that Leydig cells or interstitial cells present inside the interstitial space of the testicular lobules produces androgens or testosterone. So this statement is true. Next is oogenesis takes place in corpus luteum. No, oogenesis is a process of formation of ova and occurs inside the ovary. So this statement given, the statement E, that oogenesis takes place in the corpus luteum, is actually false statement, as oogenesis takes place inside ovary. Inside ovary. The next statement we have is menstrual cycle ceases during pregnancy. Yes, it is true statement that menstrual cycle actually ceases or stops during pregnancy. This statement is actually correct. Now next and the last statement of this question we have is presence or absence of hymen is not a reliable indicator of virginity or sexual experience. This is also a correct or true statement, that presence or absence of hymen is not a reliable indicator of virginity or sexual experiences, as hymen actually often breaks due to some sport active women. So this is not a sign of virginity or any sexual experience. So this is all the statements we have done with. The next question is, what is menstrual cycle? Which hormones regulate menstrual cycle? So what is menstrual cycle is actually, menstrual cycle is a reproductive cycle in females. Menstrual cycle is a female reproductive cycle which is characterized by the cyclical changes occurring in the endometrium lining of the uterus. So menstrual cycle is a cycle of reproduction occurring in the females, and it is characterized by the cyclical changes occurring in the endometrium lining, which is the innermost lining of the female uterus. So this is the answer of menstrual cycle. Menstrual cycle actually occurs for about 28 to 29 days. 28 to 29 days on an average. Now here we comes with the second part of this question, that which hormones regulate menstrual cycle. Basically there are two hormones known as gonadotropins and named as follicle stimulating hormone FSH and luteinizing hormone LH. So we have two hormones, that is follicle stimulating hormone and luteinizing hormone. If we talk about these hormones, these two hormones, both the hormones actually regulate the menstrual cycle. Let's see how. First of all, if we talk about the follicle stimulating hormone stimulates the growth and development of the follicles present inside the ovary. So it stimulates the growth and development of the follicles present inside the ovary. So this is the function of follicle stimulating hormone. Next, when we move ahead with the luteinizing hormone LH, this actually stimulates the maturation of Graafian follicle, which is a matured follicle inside the ovary of a female containing secondary oocyte. Now LH stimulates the maturation of a Graafian follicle, and also in between or in middle of the menstrual cycle, that is from 12 to 14th day, LH the amount of luteinizing hormone actually increases, and we call that increase of LH is actually LH surge. So we can see LH surge in the middle of the menstrual cycle. But what is the significance of this LH surge? Why there is increased amount of LH in the middle of the menstrual cycle? Now we have discussed about the basic functions of LH and FSH. LH stimulates the maturation of Graafian follicle, and in between of the menstrual cycle there is the increased amount of LH, luteinizing hormone, we call it LH surge. This LH surge actually stimulates the rupturing process, or it helps in the rupture of Graafian or matured follicle, and releases the secondary oocyte from Graafian follicle. So these are the functions of FSH and LH. Along with these functions, both of the gonadotropins such as follicle stimulating hormone and luteinizing hormone, both of these hormones actually stimulates the follicles to produce hormones, that is estrogen and progesterone, which in turn provides the environment, suitable environment for the possible pregnancy in the uterus. When after ovulation, the increased amount of progesterone and estrogen, they will give negative feedback to the hypothalamus to stop the synthesis of follicle stimulating hormone and luteinizing hormone. So this is all about the menstrual cycle and the hormones which are regulating the menstrual cycle in the females. The next question number 18 we have is, what is parturition? Which hormones are involved in the induction of parturition? So first of all, parturition is actually a process of giving birth to the young one. Parturition is a process of giving birth to the young ones. Giving birth to the young ones, or childbirth we can say. So this is the process of giving birth to the young ones. And if we talk about the hormones which are involved in the induction of parturition, there are actually two hormones which are involved in the parturition process, that is oxytocin and relaxin. So let's understand what are these two hormones actually doing in the process of parturition. First of all, at the time of parturition, which is a process of giving birth to young one, what happens? The developed fetus and placenta, which is actually placed inside the womb of the female uterus. So developed fetus and placenta sends signal for the parturition process, which results into the dilation of cervix, and this process of dilation of cervix, which is performed by the sending of signals through placenta and developed fetus, this process is known as fetal ejection reflex. So first thing we have is fetal ejection reflex, in which the cervix gets dilate. The cervix gets dilate. The developed fetus and placenta sends signal for the dilation of cervix and parturition process. This is known as fetal ejection reflex. When the fetal ejection reflex takes place, this fetal ejection reflex gives positive feedback, and this positive feedback goes to the posterior pituitary gland, from where the secretion of oxytocin happens or occurs. Oxytocin is being released from the posterior pituitary gland through the fetal ejection reflex. This fetal ejection reflex gives positive feedback on the posterior pituitary gland, from where the production of oxytocin takes place. This oxytocin hormone is mainly responsible for the process of parturition. After the formation of oxytocin hormone, it further dilates the cervix, leading to the parturition. Now, there is another hormone known as relaxin hormone, which is produced by the corpus luteum at the end of the pregnancy. Relaxin hormone is produced in the ovary by the corpus luteum at the end of the pregnancy, and this relaxin hormones dilates the pelvic wall, dilates the pelvic region as well as cervix, and resulting into the parturition process. So these are the hormones, oxytocin hormone and also a relaxin hormone. These two hormones are actually involved in the induction of parturition, and this is how parturition actually takes place inside the female. Question number 19. In our society, the women are often blamed for giving birth to daughters. Can you explain why this is not correct? First of all, this is absolutely wrong thing that females are responsible for giving birth to daughter child or female child. Why this is so? As we know that gametogenesis Gametogenesis are the two types of gametogenesis we have, that is spermatogenesis and oogenesis. In the spermatogenesis, there is a formation of male gamete takes place, means the sperm cells are made, and in the process of oogenesis, there is a formation of ovum or egg cell or female gamete. Both the gametes are actually haploid, that means both the gametes are having 23 number of chromosomes. Now, we have in a normal cell, if we talk about the 44 chromosomes we have, or we can say 23 pair of chromosomes, as we know that chromosomes are actually occur in pairs. Now, among 23 pair of chromosomes, 22 pair of chromosomes are normal, and we can call them allosomes. Allosomes. One pair of chromosome we have is sex chromosome. Now, if we talk about the sex chromosome, we have a pair of chromosome, let's say XY. XY chromosomes are the sex chromosomes, but this case is a normal case in which we are talking about a diploid cell, and diploid cell is having 23 pairs of chromosome. If we talk about the gametes, if we talk about the sperm cells and ovums, they are having 23 number of chromosomes only, among which they have 22 chromosomes as allosomes, normal chromosomes, and only one chromosome is the sex chromosome. At the time of formation of gametes, we know that let's suppose this is spermatogonia or oogonia or any of the germ cell which is going to give rise to the gamete. Now, at the time of formation of gamete, from a pair of sex chromosome, only one chromosome of sex chromosome will go into the gamete. This is a by chance factor whether the X will go into the gamete or the Y will go into the gamete. This is a by chance factor. So let's suppose if we take the male case and female case separately, let's write here male, this here we have female. We are talking about the sperm cells, and here we are talking about the ovum cell. Now, in the case of females, the females will have only XX chromosomes, XX sex chromosomes, and XY chromosomes are present in the males. Now, at the time of formation of ovum, it will receive 22 chromosomes normal, and one chromosome as sex chromosome, and that one sex chromosome in the female gamete will always be X only. This will be X only, as the normal cell of any female which is going to give rise to the female gamete is having a pair of sex chromosome containing only XX. The sex chromosome in oogonia is XX. At the time of formation of female gamete or ovum, it will receive only X. Now, in the case of male, the spermatogonia having a pair of chromosome having X and Y as sex chromosome. Now, at the time of formation of sperm cell or male gamete, it can receive, there is a by chance factor, or we can say the 50% of the sperm will receive the X chromosome as sex chromosome, and 50% of the sperm cells will receive Y as sex chromosome. Now, at the time of fertilization, it is also a by chance factor. At the time of fertilization, whether the X will fertilize this ovum containing X chromosome, or whether this sperm containing Y chromosome will fertilize the egg or ovum. If in case at the time of fertilization this egg is fertilized with, this egg is containing X already, this egg is if fertilized with the sperm cell containing X chromosome, then the zygote formed will transform into female fetus or female child. And if in case this ovum containing X chromosome already, if fertilizes with the sperm containing Y chromosome, then this zygote will transform into male child. So here, from here you can clearly see and clearly understand that who is actually responsible for the sex of the child. This is very clear from this that only the male is responsible for the sex of the child, not the female is responsible for the sex of the child, as female gamete are always having X chromosomes, and male can have both X or can have Y chromosome. So this is the answer of this question, that when the society actually blames women for giving birth to the female child, this is actually wrong, as the males are the only responsible factors for the female childs. Question number 20 is, how many eggs are released by a human ovary in a month? How many eggs do you think would have been released if the mother gave birth to identical twins? This question is basically having three parts, and the last part of this question is asking, would your answer change if the twins born were fraternal? First part of this question contains a question that how many eggs are being released by a human ovary? That is one egg. Answer of the first part of this question is one egg. Only one egg actually released from the ovary per month or per menstrual cycle. So this is the answer of for the first part of this question. Next, moving towards the second part of the question, that how many eggs do you think would have been released if the mother gave birth to the identical twins? So in the case of formation of identical twins, there only one egg is being released from the ovary. Second part of this question is having answer as one egg. Then question arises, how can one egg actually forms two childrens which are actually identical to each other? Now, what happens in the formation of identical twins? Let's see. This is ovum, which actually gets fertilized with sperm cell. We know that secondary oocyte releases from the mature graphene follicle from the ovary into the fallopian tube, where the secondary oocyte meets with the sperm cell and completing the second meiotic division and transforming into an ovum. The fertilization is being taken place inside the fallopian tube. After the fertilization, we know that there is a formation of a diploid cell takes place, that is zygote. Let's suppose this is a zygote which is 2n. This is zygote. Now, we are discussing the case of identical twins. In the formation of identical twins, this zygote, in a normal case we know that zygote tries to cleave, it actually cleaves and forms embryo further. Now, in the case of identical twins, this zygote, when cleaves into many blastomeres or different types of cells to form an embryo, what happens in this case of identical twins? This zygote, when cleaves, this will cleave into two different cells, two different cells which are diploid in nature. This zygote, when cleave, it will cleave into two separate cells. Both of these cells are 2n, that is diploid, and both of these two cells which are formed from the zygote only, which are resulted from the cleavage of the zygote only, these two cells will grow into new individuals separately, and they will they are identical to each other. Why they are identical to each other? Because they are generated from one zygote only, zygote which is formed from the fusion of one sperm cell and one egg cell. Hence, this is the reason actually that these two separate individuals formed from a zygote are actually identical to each other. So it is clear that in the case of formation of identical twins, there will be only one egg is being released from the ovary, which will transform into zygote. At the time of cleavage, the zygote will transform into two diploid separate cells, which eventually grows into new individuals which are identical to each other. Now, next part or the third part of this question is asking, would your answer change if the twins born were fraternal? First of all, what do you understand by this term fraternal? Fraternal is just another name for non-identical twins. We all have seen the identical twins or a mother which is giving rise or giving birth to identical twins, sometimes mother giving birth to non-identical twins. We have already discussed the case of identical twins. In the identical twins, only one egg is being released and the formation of zygote will transform into two different or separate cells. In this case, the two growing individuals are similar to each other, exactly similar to each other, identical to each other. Now, in the case of fraternal twins, their answer is changed. Now, C part or the third part of this question is having, if we are talking about the fraternal twins, there will be two eggs. Sometimes two eggs are being released at the same time from the ovary into the fallopian tube, and we know that there is no polyspermy occurring in the case of humans. Only one sperm cell can fertilize an egg or an ovum. So, in the case of non-identical twins, two eggs are being released from the ovary into the fallopian tubes. Both of the two eggs are fertilized with the two sperm cells, fertilized with the two separate sperm cells or male gamete, two sperm cells. And both of the zygote, as we know that fusion of one male gamete and of one female gamete will result into the formation of a zygote, and zygote will transform into a new individual. So in the case of non-identical twins or fraternal twins, what happens? Two eggs are being released from the ovary into the fallopian tubes. Both of the two eggs meet with the two separate sperms and fuses with them and forms the zygote. There will be two zygote formed in the case of non-identical twins or fraternal twins. So this is the complete answer of this question, that first of all, in the normal case, only one egg is being released per month or per menstrual cycle. In the case of identical twins, there will be only one egg. And in the case of non-identical twins or fraternal twins, there will be release of two eggs or ovum occur in the female. Now let's come towards the last question of this chapter or of this session, that is, how many eggs do you think were released by the ovary of a female dog which gave birth to 6 puppies? Now we know that generally a female dog actually gives birth to 6 or 7 puppies at a time. So all these puppies, let's say these 6 puppies are actually not identical. They are not identical to each other. They are not identical to each other. Hence, there must be release of 6 ovums or 6 eggs in the ovary of a female dog. So, how many eggs are being released? If any female dog is giving birth to 6 puppies, there must be release of 6 eggs. 6 eggs or 6 ovums which will get fertilized with 6 separate sperm cells, and hence forming the zygote, separate zygotes which eventually grow into 6 puppies. So 6 is the answer. 6 eggs will be released in the case of female dog which will eventually give rise to 6 puppies. So this is all for today. We have discussed all the questions given in the NCERT textbook of this chapter, Human Reproduction. Thank you and have a nice day.